Thank you. Thank you very much for this kind introduction. I hope everybody can hear me right. So today I'm going to talk about influence of host genetics in shaping the human bacterial community in the beef cattle. So first of all, this study would not have been possible without Dr. Samodasi Fernando, who's also a PI in our lab, and Dr. Matthew L. Spangler from University of Nebraska Lincoln. And Dr. Matthew really helped us through our GWAS analysis, and then our collaborators at a US Meat and Research Center, USDA. First of all, uh, I will talk about a little bit about this cattle microbiome. I think most of the audience here work a lot with the human microbiome, but for the cattle microbiome, a lot of studies has been done to see the cattle microbiome, like their oral microbiome, digestive tract microbiome, their reproductive tract microbiome, or skin microbiome. And the interesting thing is the Firmicutes are the major group of bacteria that we can find in most of the cattle microbiome except the skin microbiome. But for today's talk, I'm going to talk about and more focus on the human microbiome. So these cattle have this very unique digestive tract as compared to what we call these non-ruminants or the monogastric animals. So they have basically four compartments like rumen, reticulum, omesum, and abomasum. And rumen is just a mix of a lot of different microbes. If we look at the rumen at this time, they have bacteria, they have methanogenic archaea, they have ciliate protozoa, fungi, and bacteriophages. And in just 1 ml of the rumen fluid, we can find billions of bacteria and millions of all the other different microbes. And to just give you a perspective of one cow can hold up to 25 to 28 gallons. So that's their whole capacity. And in 1 ml, we find like all of these large number of different microbes. So in our animal sciences, people have studied these different human microbiomes because we talk about these beneficial microbiomes, but in cattle, cattle lives with this human microbiome in a symbiotic relationship. So in this symbiotic relationship, host is getting the functions of these microbes, like these microbes help the cattle to digest the undigestible coarse material like the cellulose, cellulase, feed materials like those fodder, forages, and in response they produce these volatile fatty acids. And these volatile fatty acids are metabolized by host animals like these cattle, and they actually depend on these volatile fatty acids to get their energy. Like 70% of their energy comes from these microbial products. And in addition to these VFAs, they also get these microbial proteins. So actually, there's a lot of fermentation is going on in the human, and at the end, hosts utilize the product of these bacterial or other microbial fermentation. So first of all, why we did this study when we looked at these human microbiome throughout the literature, a lot of studies has been done, and the major factors that are known to affect the human microbes, age is one of the major factor, then diet, or the one of the major factors. And if we have all these different factors that influence host microbiome or the host efficiency or performance, then we get the diet and host genetics also. But when we were planning this research at that time, we did not have enough evidence or the clear evidence if there is any top-down effect of host genetics on the human microbes. Most of my audience would be familiar with these the lot of studies in humans where the lot of great scientists looked at the effect of host genetics, but at that time we couldn't find anything in the cattle microbiome. So we wanted to see if there is any host genetics effects on the human microbiome. Uh, so to just give you an overview about the methodology of my research, so we have more than 500 animals. And briefly, so we have the human samples and we extracted the DNA and then we sequenced the V4 region on the Illumina MySeq. And at the end, when we process the data, we have phyla level, family levels, and operational taxonomic units or the bacterial species level data. And from the same animal, we also collected their blood samples, and we genotyped these animals based on these SNP arrays. So these SNP arrays are basically one nucleotide differences in the genomes of different animals. And at the end, we have more than 61,000 SNPs. And then we use the SNP data and then OTU family and Phyla the microbiome data, and we run the GWAS analysis to see if we could find any associations between the host genetics and the human microbiome. So to just give you an overview about our results, so on the x-axis we have this posterior mean heritability. So this is the how much genetic control the host genetics have on the human microbiome, and on the y-axis we have the number of different OTUs or phyla. For example, if we looked at the phylum level, so 0.25 is represents like the very can say low genetic control or low heritability, and as its number approach to one, it means like very high genetic control. So there were like 3 to 4 phyla that has the heritability more than 0.25, and same was true for family and the OTU levels. And when we looked at some of our the highly heritable phyla, so we have one phyla that has the really high heritability. And then same was true for we have a couple of families with the high heritability and the OTUs, a lot of different bacterial species. And when we looked at our all these heritability data, we see uh, at phyla family and OTU level, we have some members of the human microbiome with the very high heritability. Just due to we have uh, uh, small time, little time, so I will focus on one example. For example, this methane producing archaea. So we have this family Methanobacteriaceae, and then the member of this Methanobacteriaceae, the OTU 41 Methanobacter, and they have like really high this heritability. And these organisms are actually known for the methane producing organisms in the cattle. As we know, with all this global warming and the climate change, so we want to mitigate the effect of methane. We want to produce and control methane. So this was really surprising that host genetics is actually have effect on this methane producing archaea in the human microbiome. So after looking at these individual microbiome or the individual members of the bacterial species. So then we actually looked at the 1 MB region. So in this table on the first is showing the class like OTU family or the phyla level, and then we have chromosome. So basically what we did is instead of looking at the individual SNPs, so we looked at the 1 MB region. So megabase on different chromosomes, and then we add the effect of all the SNPs on the megabase region. And this graph actually represents the top 7 to 8 megabase regions that have the associations with the OTU family and the phyla level. So in this, this column is the position. So the MB start to end represents. So this is, for example, chromosome number 1. Megabase starts from 132 MB region to 133, and then we have these different OTUs. And same was true for the family and phyla. To just give you an overview, but we find different patterns. For example, we saw the same chromosome and the same region have the association or effect at the family, OTU, and the phyla level. We actually looked at if the member of these OTUs were actually member of either these phyla or these family. Two of these OTUs were actually member of either RF16 family or the phyla Fibrobacteraceae, but this OTU 19 that was from bacteroids. So we can say there are some OTUs that have the associations because there are part of the bigger family or phyla that also have genetic control, but there were some OTUs that were individually have association with the host genetics. And then there were some OTUs, like the similar OTUs controlled by two different regions, like these OTUs 30 and 2337. So this graph actually we then plotted all of these chromosomes to the scale. So these chromosomes 1 to 29, and the main objective of plotting these graphs was to look at these 1 MB region. So this red region is actually the 1 MB region that I talked about in the graph. And we also then looked at, okay, can we find any genes on those regions that have the association with these human microbes? And when we looked at, we did find some genes present on those regions, but for the time being, I will just focus on one example. For example, this chromosome number 1 have this region that have the association with different microbes. And if we looked at this region, it also has this claudin 18 protein. And then we looked at the what kind of microbes are associated at this region. So there was one microbe, the succinoclasticum, and in human they play a very important role to produce these volatile fatty acids from succinate to propionate. And we hypothesize that it's possible that host is selecting for different microbes that are helping them to produce more VFAs and then have some genes on the same regions. Like these claudin 18 proteins are the part of those junction proteins in epithelial cells that play their role to kind of control the movement of solute and the water across those barriers. To just summarize, we did see the effect of host genetics on OTU family and the phyla level, and then there were some identified regions that we see they might be regulating the abundance of certain microbes, and then there were we think the host is also selecting for the microbes to increase the volatile fatty acids to fulfill their energy needs. And at the same time, there are some genes present on those regions that may help those volatile fatty acids to increase their absorption. At the end, when we were looking at all of these microbes, we were more focused on, okay, can we find like actual their functions or what these microbes are actually doing? But the biggest constraint for these microbiome study, especially is true for our cattle microbiome, we don't have really good idea about the functions. A lot of the OTUs were not identified at the species level, and then we were not sure about, okay, what actually functions they are performing. So for that reason, this project is actually in the second phase, where we are now working on the metagenomes. So we are going to follow the same concept, but next time we are going to be more focused on the shotgun sequencing, and hopefully we can find the functions of these microbes to have better understanding how these hosts, on the one hand, have these top-down effect on the human microbes, or actually what the functions of those human microbes are to better understand the cattle microbiome. So with that, I would like to take any questions.